All right, so now we're moving on to oxidative phosphorylation. So if you've watched the previous few mini lessons, we've gone through glycolysis and citric acid cycle, and now we're going to look at the final step of cellular respiration. So this oxidative phosphorylation is also occurring in the mitochondrium of the cell. And just like all the other steps, if one of the other steps is interrupted before oxidative phosphorylation takes place, then oxidative phosphorylation cannot take place. Okay, so if the citric acid cycle somehow fails, or if pyruvate oxidation fails, or glycolysis has failed, then oxidative phosphorylation won't take place. So this is a series of events. So oxidative phosphorylation includes something called the electron transport chain, as well as chemoosmosis. So in this process, remember all of the NADHs and FADH2s that have been formed through glycolysis and pyruvate oxidation and citric acid cycle? Well, each one of those electron carriers have made their way into the inner folds of the mitochondrium, the cristae, and they are going to finally release those electrons, and those electrons will get picked up by the electron transfer chain. Okay, so the electrons that were shuttled by NADH and FADH2, they're going to end up being dropped off in the electron transport chain. Okay, so this first step of oxidative phosphorylation takes place with both the inner membrane of the mitochondrium, the mitochondrial matrix, as well as the outer membrane, or the outer space, or the inner space, I'm sorry, between the inner and outer membrane. So the folds, I should say, of the cristae of the mitochondria. Okay. So the carriers, remember the NADH, NAD+, those alternate between reduced and oxidized states as they accept and donate their electrons. So when we have NAD+ accepting those electrons, okay, it's going to be reduced to the form NADH because it's gained those electrons. And then those electrons, once they are released from NADH, okay, the NADH loses those electrons and becomes oxidized to NAD+. Okay, so this is, again, that redox reaction. So electrons are transferred from the NADH and the FADH2. Those electrons, when they get released, they're going to be transferred to the electron transport chain. And then those electrons are going to be passed through some proteins that are embedded in the cristae, the inner membranes. And those are just called cytochromes, which each contain iron atoms. And finally, the electrons are going to end up to the final electron acceptor, which is our oxygen molecule. Okay, so electrons drop in free energy as they go down the chain, and they finally get passed to our electron acceptor, the oxygen molecule, and to form water. So water will be the end product there. So how does that occur? Okay, so let's go back to our electron carrier NADH. So notice NADH has those electrons attached. And then we have oxidation take place. So the NADH loses those electrons and becomes oxidized to NAD+. And those electrons start becoming part of this big protein that's in the membrane of the inner membrane of the mitochondria. So watch the electrons. So the electrons are going to pass through the proteins, or the protein complexes. Now, as they're being passed through, eventually they're going to end up becoming part of oxygen as the final electron acceptor. So that oxygen molecule is going to gain those electrons, it's going to be reduced, right? So what this process is doing is it's causing hydrogen to move across the membrane, okay? So the hydrogen ends up getting pumped across the membrane as these electrons are flowing through, okay? So the movement of hydrogen ions across the membrane will eventually allow for those electrons to be dragged through, so it's helping each other. And eventually we're going to end up harvesting those energy to form water as well as ATP. And we end up with a lot of ATP in this process. In fact, around 32 ATP. So let's go ahead and look at the details. So here is our electron transport chain. You can see that the NADH is carrying the electrons. They get released. As they get released, this is causing a protein complex to change to allow hydrogen ions to flow across from the mitochondrial matrix into the inner membrane space. Okay, so because those electrons are traveling through, it opens this, what we call this proton channel. Okay, so now hydrogen ions are getting pumped across. So if you look at where the electrons are going, they're passing through the inner membrane here. And as they pass through, they're passing past some different protein complexes. And eventually they're going to be accepted by the oxygen atom to form water. So what effect would an absence of oxygen have on the process illustrated below? So what if we don't have oxygen? No oxygen. What happens? Well, if there's no oxygen, where would the electrons go? So if there's no oxygen, then there's no electron transport chain because there's no place for the electrons to move to, okay? So they're not going to be able to be accepted anywhere. And so the whole process will shut down. And if there's no electron transport chain, then we cannot get any ATP. So electron transfer in the electron transport chain causes those proteins to pump hydrogen ions from the mitochondrial space to the inner membrane. Okay, so let me just go back. Oops, let me go back a slide. So while those electrons are moving through the electron transport chain, it's opening proton channels. So now all of the protons on the mitochondrial membrane space start flooding across the membrane into the inner membrane space. That inner membrane space is getting flooded with hydrogen ions. There's a lot of hydrogen ions there, okay? So those hydrogen ions are then going to pass through an enzyme called ATP synthase. And as they pass through that enzyme, let me go back a slide, show you where I am. So here's ATP synthase over here. All of these hydrogen ions, they're going to pass through this enzyme. And as they're passing through this enzyme, they're releasing enough energy to combine ATP and phosphorus together to form ATP. Okay, so that this flood of hydrogen ions through this enzyme is going to provide energy to bring together ADP and phosphorus to form ATP. Okay, so ATP synthase is the enzyme that's involved, or that's going to funnel the hydrogen ions through. Okay, and it creates this exergonic flow of hydrogen ions to drive the phosphorylation of ATP. Okay, and this is also known as chemoosmosis, this step, because it uses energy in a hydrogen ion gradient to drive cellular work. Because we have a lot of hydrogen ions on one side of the membrane, the inner membrane space, as it's flooding through, okay, you know, there's a gradient forming. We have a lot of positive ions on one side of the membrane, a lot of negative ions on the other. And so that's going to create this hydrogen ion gradient. Okay, and that's going to be used to drive cellular work. So here's a picture of a very close-up view. So here's the cristae, okay, the folds, the intermembrane folds of the mitochondrium. Here is the mitochondrial matrix, okay, and then here's the intermembrane space up here, okay. So remember, we have over on the left-hand side, imagine the electron transport chain going on. And so all these hydrogen ions get pumped across into the intermembrane space. And now we have all these hydrogen ions up top in the intermembrane space, and they are going to get funneled through this complex enzyme called ATP synthase. And as they get funneled through, we got some energy being generated to bring ADP and organic phosphate together to form ATP. Okay, so now we got lots of ATP being generated. So that's bringing it all together. So we're taking the close-up view and expanding it back so we can see the electron transport chain. Those hydrogen ions get pumped across. We now have a huge gradient of hydrogen ions on one side of the membrane. And so ATP synthase will open the channel. Hydrogen ions will flood into the mitochondrial matrix, providing an exergonic reaction to occur, providing the energy for phosphorylation to take place. So just a review of the whole process. We started out with glycolysis that occurred in the cytoplasm of the cell, where we had glucose, our initial input of glucose, forming 2 pyruvate molecules. And in the process, we netted 2 ATP and 2 NADH, okay? And then from there, we those 2 NADHs traveled towards the inner membrane into the cristae, that's where they got dropped off in that inner membrane in the electron transport chain. Ignore this FADH, by the way. Then we have pyruvate oxidation take place, with the pyruvate being oxidized to form 2-acetyl-CoA and Na2-NADH being basically gaining those electrons, and so that will be moving towards the electron transport chain. The citric acid cycle will further break apart acetyl-CoA or acetic acid and forms 2 ATP in the process, 6 NADH and 2 FADH2s. So now we're combining everything from those processes, the NADH from glycolysis, the NADH from the pyruvate oxidation, the NADH and the FADH2 from the citric acid cycle, all shuttling electrons to oxidative phosphorylation transfer. And that's going to end up giving us enough electron force to bring together ADP and the phosphate groups to form anywhere from 26 to 28 ATP. So if we are adding the ATP that's acquired from glycolysis plus citric acid cycle plus the oxidative phosphorylation, we end up with about 30 to 32 ATP on average generated from one single glucose molecule. Okay. So this is a good summary of the entire process. So we start with glycolysis. You can see the inputs, the outputs, and then moving on into pyruvate oxidation, citric acid cycle, and then finally oxidative phosphorylation.